Hi everyone! We don't have an online lecture this week, so I'm kind of taking advantage of that to make sure we really cover some of that material from respiratory physiology that we didn't get to during the in-person lecture. So if you're in the Monday section, then you've already gotten through the bulk of the respiratory physiology lecture. If you're in the Friday section, I'm going to get to a stopping point in lecture on Friday, and then we'll get to this material online. I also wanted to say that because we're getting to the endocrine material, kind of close to the actual exam, I want to make sure you have time to remember and quiz yourself on a lot of the functions of different hormones. So I'll be posting that material today as well. Okay, so we talked in previous lectures about the relative structure of erythrocytes, of hemoglobin. We talked about their functional role. Now we're going to be integrating the cardiovascular system, the respiratory system, and that structure of hemoglobin within erythrocytes to think about this idea of oxygen saturation and how we have different carrying mechanisms for carbon dioxide and oxygen. We're also thinking about the factors that affect that carrying of oxygen, so how readily hemoglobin both hangs onto oxygen and also gives it up to our body tissue. So we'll think about something called an oxygen-hemoglobin dissociation curve. There's this idea of shifting the curve one way or the other. This is that part of the lecture. So remember when we learned about blood physiology, we talked about hemoglobin, we talked about whether blood is oxygenated or deoxygenated, and there's a common misconception that deoxygenated blood is blue in color. Oftentimes because we look at our systemic veins, they appear blue, but that just has to do with the diffraction of light. So both oxygenated and deoxygenated, quote unquote, blood is red in color. It's just kind of different shades of red. When we think about pulse oximetry and actually measuring oxygen saturation, that light difference, that color difference is what we actually take advantage of to do that in a clinical setting. So with this oxygenated blood, what we have a really high proportion of is oxyhemoglobin. And we use these terms oxygenated and deoxygenated, but blood is never fully deoxygenated. There's almost always oxygen still in the blood and still bound to hemoglobin. So it's really just a matter of what proportion of that, what the oxygen saturation is. So when oxygen is bound to hemoglobin, we call that oxyhemoglobin. When oxygen is not bound to hemoglobin, we call that deoxyhemoglobin. And when instead carbon dioxide is bound to hemoglobin, we call that carbaminohemoglobin. We'll get back to that in just a moment. I just wanted to remind you that oxygen binds to hemoglobin a lot more readily than carbon dioxide does. Carbon dioxide can be carried in a lot of other ways within our body. So when we're thinking about that overall structure of hemoglobin, remember that we have those 4 globin subunits, so those different proteins that come together to the bigger hemoglobin. There's a central heme, which is a red pigment that has a central iron ion. So the heme is central in all of those, and then there's an iron ion within each heme. In a previous lecture, we talked about how this is broken down. Just remember that with all of the hemoglobin in all of the red blood cells in your body, you have a tremendous capacity to carry oxygen. So again, a key function of erythrocytes, of our red blood cells, is carrying respiratory gases. So when we're thinking about that carrying and thinking about oxygen binding to hemoglobin, remember that there's these conformational changes that allow oxygen to more and more readily bind to hemoglobin. That first oxygen binding to the first subunit requires a little bit of time, but as we have those conformational changes, oxygen binds more and more readily. And when all 4 hemoglobins, or all 4 globin proteins with a central heme within the single hemoglobin have oxygen bound to them, we say that that hemoglobin is fully saturated. So when we're measuring oxygen saturation of hemoglobin, we're thinking about what proportion of total hemoglobin in our body actually has oxygen fully bound to it. So that idea of oxygen saturation, again, is the percent of total hemoglobin that are saturated with oxygen. For us, in like a healthy adult, on average, it's anywhere from 95 to 99 or 100%. in a previous lecture, I talked about how we actually go about measuring this, but you should know that there's a relationship between that percent saturation of hemoglobin and also the partial pressure of oxygen. So we see that in this diagram over here. So right here, this is the alveolus. This is a pulmonary capillary. We have a lot of oxygen right here and a much lower partial pressure of oxygen down here in the pulmonary capillary. And part of the reason why we have that strong concentration gradient is because as that oxygen moves from that higher partial pressure of oxygen to the lower partial pressure of oxygen, it is immediately bound to hemoglobin. So if we have a lower percent saturation of hemoglobin through those pulmonary capillaries, that's going to bind to the oxygen that's coming in from the alveolus much more readily. So we have this immediately picking up that oxygen and maintaining a strong concentration gradient, which keeps the oxygen flowing in. So when we're calculating that oxygen saturation of hemoglobin, we measure that as a percentage. You might also see this referred to as SO2. That's referring to the saturation of oxygen, like how PO2 is referring to the partial pressure of oxygen. So again, we're going to take the fraction of saturated hemoglobin and divide it by the total amount of hemoglobin, both unsaturated and saturated, and then we just multiply it by 100 to get a percent. So just to kind of visualize this, that unsaturated hemoglobin does not have oxygen bound to those heme groups within the globin proteins. The saturated hemoglobin do. So a couple of weeks ago, we also thought about the idea of cardiovascular centers in terms of hemodynamics and regulation of the cardiac cycle. So these are responding to a lot of different factors, including chemical changes. So they can respond to pressure changes, to chemical changes, all sorts of things. But in part, they monitor the pH as a proxy for cellular activity. So the reason for that is if you have a very active cell, the environment is going to be low in oxygen, high in carbon dioxide, and has a low pH. The reason for that is because oxygen is used up by cellular respiration, carbon dioxide is produced. Carbon dioxide mixes with water to form carbonic acid, which dissociates into bicarbonate ions and hydrogen ions. And then you have these free hydrogen ions that lower the pH, making it more acidic. So a low pH indicates cellular activity. A high pH indicates a cell at rest. So in that case, you would have high oxygen. The thing that is directly affecting the pH is that low CO2 because there's less carbon dioxide being produced through cellular respiration, and there's a higher pH, so it's less acidic. So we learned this already, and you have this kind of innate knowledge that a more acidic condition is associated with a more active cell. And so we're going to think about that, and we'll get back to that in just a moment in terms of the oxygen hemoglobin dissociation curve actually shifting. So when we look at this at a kind of state of rest, kind of like your just average cell activity, we see this S-shaped curve. On the x-axis, that is the partial pressure of oxygen. And then on the y-axis, that's the oxygen saturation of hemoglobin. So we know that there's this relationship between partial pressure of a gas and how much of that gas is available to saturate hemoglobin. And so there's a few different parts to this curve. There's this kind of like straight vertical portion, which represents the range of partial pressure of oxygen within the systemic capillaries. There's also a more flat portion up here, which represents the range in the pulmonary capillaries. So that difference is kind of the volume of oxygen that is unloaded from the oxygen-rich blood that's coming from the pulmonary capillaries into body tissue. So we kind of have this, again, innate understanding that if there's a higher partial pressure of oxygen, there's going to be more oxygen saturation. If you think about the pulmonary capillaries, those are right next to the alveoli, which are bringing in oxygen-rich air from the external environment. Of course, there's a higher partial pressure of oxygen there, and so there's going to be a higher oxygen saturation of hemoglobin. So when we think about the idea of the curve shifting, right now, this graphic right here is just showing you the direction of the shift. I'll show you how the shape of the curve remains pretty constant, but we actually get like the whole curve moving. And I'll try to find some videos that more visualize this. I tried to find GIFs and there really weren't any about like showing that shift of the curve. So I'll post more resources about this. But it turns out that those factors that represent a more metabolically active cell, the partial pressure of carbon dioxide increasing, the kind of concentration of those protons, which represent a more acidic or environment or lower pH, a higher temperature because heat is produced through cellular respiration, all of those shift the curve to the right, which means that hemoglobin gives up oxygen a little bit more readily. So there's a decreased affinity for oxygen. That means that it doesn't hang onto it as much and it gives it to our cells more readily. If there's a left shift, if the whole curve physically shifts to the left, that happens in cases where there's less carbon dioxide, there's less protons being produced, so a higher pH. There's a lower temperature. This happens with fetal hemoglobin as well, which we'll get back to in just a moment. So in those situations, there is a higher affinity of hemoglobin for oxygen, meaning it hangs onto it a little bit more and doesn't give it up to our tissues as readily. So here we see this 2,3-DPG. Just so you know what that is a molecule that fits into deoxyhemoglobin, and it helps maintain the shape. So if there's more of that, then the hemoglobin is going to give up oxygen a little bit more readily because it's preferentially binding to that 2,3-DPG. So that's involved in fetal hemoglobin as well, and we'll talk about it in terms of altitude sickness at the end of the lecture. So thinking about shifting the curve, if you look at this graph, it's showing the effect of pH and how that whole curve, that whole dissociation curve, physically shifts in different pH environments. So for the same partial pressure of oxygen, let's say about 40, if you're at these lower pHs, which represent more cellular respiration, for the exact same partial pressure, there's going to be less of these hemoglobin molecules that are fully saturated with oxygen. If you're in a kind of cell at rest or kind of just like chilling, going about normal activity, that same partial pressure of oxygen is going to have more hemoglobin fully saturated, meaning they're hanging on to it and not giving it up as readily. And if you're really at rest, if you have an even higher pH, maybe even getting into alkalosis, you have way more hemoglobin fully saturated and you're really not giving up that oxygen to your tissue, which can also be pretty bad. So all of these are kind of these physiological adjustments to maintain homeostasis, to make sure that if you need more oxygen, hemoglobin gives it up. So an active cell produces carbon dioxide through cellular respiration. Carbon dioxide and water produce an acid which lowers the pH. That sends a signal. That curve physically shifts to the right, which means that there's a lower affinity of hemoglobin for oxygen. So hemoglobin has a tendency to give up oxygen more readily to tissue during a lot of cellular respiration, and we call this the Bohr effect, this idea of the curve shifting, of the chemical properties of hemoglobin changing when we're in those different pH ranges. And I just wanted to clarify, this is like a chemical idea. It's how hemoglobin physically works. We don't rely on a signal to our brain to actually like make hemoglobin let go of oxygen. It's just how things happen. Those signals kind of affect our heart rate as well as our respiratory rate. So we see something similar with temperature for the same partial pressure of oxygen. if you're at very different temperatures, the hemoglobin will hang on to oxygen very differently. So if you're at a very warm temperature, 37 to 38 up here is kind of normal body temperature. So at that body temperature, you're giving up hemoglobin, or sorry, hemoglobin's giving up oxygen fairly readily, but still like kind of maintaining it. If you're a very active cell and very hot, you are needing a lot of oxygen because you're presumably going through a lot of cellular respiration, and so that hemoglobin is going to let go of the oxygen. And if you're in extremely cold environments, hemoglobin is not going to give, so this is the temperature of the person themselves, not the external temperature. But if you're in those extremely cold temperatures and your body temperature drops that low, you're really hanging on to oxygen and you're not going through cellular respiration as much. When people get severe injuries or when they're like drowning in cold water, oftentimes the cold water serves to kind of preserve them and prevent them from dying. People can hold their breath way longer in cold water and also survive like really serious, like almost fatal injuries just by hanging out in that cold water and dropping their body temperature down so low that they're really not going through cellular respiration and their hemoglobin's really hanging on to oxygen. So an active cell produces heat through cellular respiration. The temperature of your body, your core temperature, is raised. That curve shifts to the right, meaning there's a lower affinity of hemoglobin for oxygen. It's hanging onto it much more readily. It's giving it up much more readily. Lower affinity is giving it up. And then hemoglobin gives up oxygen to tissue when the tissue is doing a lot of cellular respiration. So earlier I mentioned fetal hemoglobin. So remember that when there's a fetus developing, they're sharing their circulatory system with the carrying parent. And so that fetal hemoglobin actually has a much stronger binding affinity for oxygen than adult hemoglobin does. That means that even if there's not a very strong concentration gradient between the fetal cardiovascular system and the maternal cardiovascular system, the hemoglobin still snatches up that oxygen real quick. So that overcomes that lack of a partial pressure gradient. So if we look at the oxygen hemoglobin dissociation curves for fetal hemoglobin and adult hemoglobin, fetal hemoglobin is farther to the left. That indicates higher binding efficiency or affinity, and so the fetal hemoglobin is going to snatch up the oxygen way more easily than the adult hemoglobin does. So that was thinking about oxygen being carried by hemoglobin, but carbon dioxide does things a little bit differently. So it dissolves much more readily in the blood than oxygen does. That's how we start getting those chemical signals that, there's this production of bicarbonate, and that's what we call the bicarbonate buffer system that changes the pH of the blood, and about 85% of carbon dioxide is actually carried as part of this bicarbonate buffer system. About 10% is bound to different plasma proteins or maybe hemoglobin if it's carbaminohemoglobin. The remaining 5 to 7% are just dissolved as gases into blood plasma. So when we're thinking about that bicarbonate buffer system, this slide is similar to one that you saw during the digestive system, digestive physiology, when we talked about the function of those parietal cells lining the stomach. So we talked about the function of carbonic anhydrase, or Ca. That's an important enzyme that actually catalyzes that formation of carbonic acid from water and carbon dioxide. That change in chemistry is something that kind of happens naturally, but remember, enzymes always speed things up, so it happens a little bit more quickly with carbonic anhydrase. It then dissociates to bicarbonate and proton, which lowers the pH. So we also see something called a chloride shift. That's when you have bicarbonate accumulating and leaving the red blood cells. It actually gets exchanged with chloride ions. So you have this movement of chloride ions. When this happens in the parietal cells lining the stomach, that's how you get a very acidic environment within the lumen of the stomach. You have those protons and those chlorides, so you effectively have hydrochloric acid. So when you're in the pulmonary circuit, this is describing the systemic circuit. When you're in the pulmonary circuit, you shift back from bicarbonate and proton to carbonic acid to carbon dioxide in order to breathe out that carbon dioxide. So the air that we breathe out has a higher partial pressure of carbon dioxide than the air that we breathe in part because we have that reverse reaction. So again, this slide is just kind of summarizing within the context of the red blood cell how the carbon dioxide is carried, and so it shows you that carbaminohemoglobin. Okay, so getting into how we regulate these different processes, we're going to kind of integrate that idea of blood physiology back with respiratory physiology and just touch base on the homeostasis of this one more time. So remember, we talked about different types of breathing, and here we're going to focus on hyperpnea. Remember that PNEA tells you that it's a type of breathing. So for example, apnea is not breathing, eupnea is quiet breathing, and hyperpnea is forced breathing. Your diaphragm, your intercostal muscles, and your accessory muscles are all involved in forcing this breathing. So hyperpnea happens in response to an increased oxygen demand. So you're increasing both the depth and the rate of your ventilation. This is important if you're in certain disease states, as well as if you are exercising, like Chris Traeger from Parks and Rec over here on the right. So you do go through this process of hyperpnea in response to increased need. It's when you have changes in your blood partial pressure of oxygen, carbon dioxide. So because you're kind of like doing this in response to those changes, your overall balance doesn't fundamentally change. Your chemistry doesn't change because you're just bringing things back to normal. And we can contrast this with hyperventilation, which is like a really exaggerated response that isn't necessarily keeping us in homeostasis. It's moving us out of homeostasis. So carbon dioxide is removed from our systemic blood very rapidly. We're breathing it out. So it is fundamentally changing that partial pressure of carbon dioxide away from normal, moving us out of homeostasis. It's not necessarily in response to a need. So hyperventilation could be due to like a panic attack, different mental states. It could be due to like different serious conditions. So it's not necessarily something that you want to do regularly unless you've been hypoventilating and now you're trying to bring yourself back to normal, and vice versa. When you hyperventilate, the treatment is to breathe slowly in and out of a paper bag so that you're forcing yourself to hypoventilate. So when you hyperventilate, you have decreased partial pressure of carbon dioxide, you have increased pH, and it's more alkaline. Sorry, that's describing the hypoventilation. So Nope, it for sure is describing hyperventilation, sorry. So hyperventilation does, because you're breathing out carbon dioxide much more quickly, you're getting rid of it, that's serving to increase your pH and making you more alkaline. Okay, when we're thinking about what's actually happening with the oxygen in our blood, there's a couple of different metrics we can use. Hypoxia specifically refers to a decrease of oxygen in your tissues. And so when you're hypoxic, you may or may not be experiencing hypoxemia. Hypoxemia is when you have very low oxygen in your blood. That's considered less than 95% oxygen saturation of hemoglobin, or the partial pressure of arterial gas oxygen between 80 and 100 millimeters of mercury. So if we were in class, I would have you kind of think about why hypoxemia is easier to measure than hypoxia. The reason for this is it's very hard to stick an oxygen probe into your tissue, but we can really easily either look at the oxygen saturation of blood using a non-invasive probe, or we can even take a sample and measure arterial gases directly. So hypoxemia in the blood, if you have that state, you for sure are experiencing hypoxia. If you're experiencing hypoxia, you may or may not be experiencing hypoxemia. So hypoxemia is a much more definite and conclusive and easy to measure way of determining deficiencies in oxygen in your body. So we're going to be thinking about this idea of hypoxemia and how our body monitors it and responds to it in terms of high altitudes. So when we talked about the bends, we talked about this idea of very high pressure. With altitude sickness, that is very low pressure. So moving up in altitude actually decreases atmospheric pressure, which also lowers the partial pressure of oxygen and causes something called altitude sickness or acute mountain sickness, aka AMS. So the symptoms of this include nausea, vomiting, fatigue, lightheadedness, drowsiness, feeling disoriented, increased pulse, and nosebleeds, and it can even cause pulmonary or cerebral edema. So if you ascend a mountain too quickly, you have this whole range of really very serious consequences, different symptoms. So when we're experiencing that, our body actually has different compensatory mechanisms to deal with that change. One of them is that there is a shift and oxygen is released more easily into tissues. There's also increased production of 2,3-DPG. So remember, when that happens, when you have more of that 2,3-DPG, then the curve is shifting to the right. So the oxygen is having a lower binding affinity for hemoglobin and vice versa. So oxygen more easily is given up from that hemoglobin. So we produce more of that 2,3-DPG to bump off the oxygen, and the 2,3-DPG binds to the hemoglobin instead. So a more kind of broad physiological process that we have is this idea of acclimatization. And so that happens when you take the time to do a long-term adjustment to the high altitude. For example, if you get to different base camps or different stations along the way of your ascent, you can take that time to acclimatize and to make sure that your body is changing its hormones, changing its production of red blood cells accordingly. So basically what happens is your body measures hypoxemia. It measures that low oxygen in your blood. That change is sensed by the liver and the kidneys, which send a signal to increase, or which they themselves produce more erythropoietin, especially your kidneys. So you have this erythropoietin, which is a hormone that signals the production of red blood cells. And so that sends a signal to the red bone marrow to increase erythropoiesis, increase the amount of red blood cells. So now, even though you're not necessarily shifting the curve, you have more red blood cells to carry oxygen. So you have increased oxygen transport. And so this is a negative feedback loop. As those oxygen saturation levels rise, EPO secretion falls. So that kind of brings things back to normal. So that is the kind of making up the lecture. Please make sure that you have that third quiz done, and I'm going to post another quiz and a reflection. So please make sure you're staying up to date on all of that work in preparation for the lecture exam.